Hi, my name is Michele Trott, and today I will be highlighting 5 publications that used Tunable Resistive Pulse Sensing, or TRPS, to characterise the physical properties of nano-sized particles. TRPS is a technique which applies the Coulter principle on a nanoscale. The Coulter principle is based on the detection and measurement of changes in electrical resistance, produced by a particle suspended in a conductive liquid traversing through a small aperture. TRPS is applied where particle size and concentration are important, such as for studies of aggregates and as part of extracellular vesicle characterization work. For suitable sample types, zeta potential measurements can also be obtained, allowing TRPS to provide information about particle stability. For more detail on how TRPS works and the factors that make it such a precise technique, follow the links below. For now, I will move on to highlighting 5 papers that used TRPS as part of their analysis. The studies address the effect of storage conditions on EVs, the development of a virus-like particle vaccine, process development for virus purification, the variability of EV characteristics in health and disease, and the quality control of submicron biopharmaceuticals. In early 2022, a systematic study was published describing the effect of storage conditions on EVs stored under different conditions. The study involved EVs from fresh, purified, platelet-free plasma and EVs derived from cell culture. The effect of slow freeze-thaw versus snap freeze-thaw cycles were compared, and possible fusion events were investigated. TRPS measurements revealed a significant reduction in EV concentration between the first and second freeze-thaw cycles, and a cycle-dependent increase in particle size for both snap and slow freeze-thaw cycles. Zeta potential was also measured to gauge particle surface charge and EV colloidal stability. As TRPS measures EVs on a single particle basis, rather than providing a bulk measurement, zeta potential data of single particles can be expressed as a size versus charge plot. The group reported a shift of the particle population towards positive zeta potential values when plasma-derived samples were stored as isolated EVs. In contrast, this change was not observed when EVs were recovered from frozen plasma samples, suggesting a protective role of plasma in this context. These changes would not be easily detectable with ensemble techniques, and here TRPS helped identify best practices related to handling and storage. TRPS measurements have also contributed to the preclinical development of a vaccine against the SARS-CoV-2 virus. A group of researchers based in Turkey developed a virus-like particle vaccine candidate with the aim of reducing our reliance on spike protein-based vaccines while potentially broadening the breadth of the immune response. In the study, they explored a few options, including several different adjuvants. Respective candidates were characterized using several techniques, including TRPS, which enabled the group to demonstrate that assembled virus-like particles were similar in size to the authentic virion. A virus-like particle vaccine developed from this study has since advanced to a phase 1 clinical trial. To explore ways to improve productivity, robustness, and control, the use of a continuous chromatography step was applied to the intermediate purification of an oncolytic adenovirus. This group tested the stability of the system under a range of different scenarios which could potentially impact virus recovery and stability. Here, TRPS was used to obtain particle size distribution of the oncolytic virus preparations, providing detailed characterization data alongside electron microscopy and measures of impurity reductions. To explore the potential use of milk-derived EV microRNA as biomarkers for subclinical mastitis in dairy cows, Switzerland-based researchers evaluated the daily stability of EV size, concentration, and microRNA cargo in raw bovine milk sampled over 3 consecutive days. The study aimed to gauge the variability of particle characteristics via TRPS, as well as microRNA profiles from isolated EVs, and compare them between cows with healthy or inflamed udders. By getting a better idea of baseline variability in healthy cows, the the group are now in a better position for future studies of subclinical mastitis biomarkers. As part of rigorous safety and immunogenic assessments of biopharmaceutical products, regulatory agencies encouraged the characterization of submicron particles. As a method suited to the analysis of samples with heterogeneous particle size distributions, TRPS capabilities were applied to the quantification of monoclonal antibody preparations. Samples were prepared by applying stir stress to IgG monoclonal antibodies to induce aggregation, and were filtered through a 5-micron membrane filter to remove large aggregates that would block the nanopore. A spiking in approach was applied to introduce an electrolyte concentration sufficient for stable measurement conditions. The authors report a novel approach to data analysis for such proteinaceous samples and describe TRPS as a promising technology for submicron analysis of biopharmaceuticals. Overall, TRPS brings in-depth insights into particle characteristics across a range of disciplines. Particle size distribution can be achieved for nanoparticles in the size range of 40 nanometers to 11 microns. And single particle zeta potential measurements can be achieved for particles suspended in TRPS-compatible electrolyte solution. If you are interested to see how TRPS can complement your nanoparticle characterization work, be sure to get in touch.